Hello everyone, welcome to my channel Bani's Biology. Today we will learn about the base excision repair that is called BER. BER. So BER is a DNA repair pathway that removes small non-helix distortion or non-bulky lesions such as oxidized, deaminated or alkylated bases and replaces them with the correct nucleotide to maintain genome stability. So it acts like a precision editor. So here by the deamination process, cytosine is converted into uracil. So deamination means removal of amino group. So here 4 position present amino group that is removed by the deamination process. So normally uracil is not present in DNA. So normal cytosine pair with guanine. So after deamination of cytosine, uracil bind with adenine that causes mutation. So then deamination process of guanine result in the formation of xanthine that bind with cytosine. And also through deamination process, adenine is converted into hypoxanthine, where hypoxanthine bind with cytosine that also causes mutation. So then by the oxidation process, guanine changes into 8-oxoguanine. So normally guanine base pair with cytosine. Then after oxidation of guanine, 8-oxoguanine base pair with adenine. For example, here a normal guanine base pair. This is the guanine base pair that normally bind with cytosine in DNA. So a normal guanine bases in DNA is oxidized by radioactive oxygen species, and this guanine convert into 8-oxoguanine. During replication, 8-oxoguanine mispair with adenine instead of cytosine. So this mispairing can lead to guanine that bind with cytosine to thymine is to adenine. This is the transversion mutation process if not corrected. So here base excision repair can remove OXO-G before replication. So if replication occur before repair mechanism, a fail-safe glycosylase remove mispair adenine, and this allow the cell to insert correct base pair that is cytosine and preventing mutation. So that's how single bases is changes by oxidation, deamination and causes DNA damage. So this damaged bases is repaired by the base excision repair mechanism. In base excision repair mechanism, first enzyme is DNA glycosylase. So this enzyme first recognize and remove the damaged bases by hydrolyzing the glycosidic bond. So where the glycosidic bond is present, so this is the incorrect bases and this is the deoxyribose sugar. So 1' carbon on the deoxyribose, this is the 1' carbon that is connected to a nitrogenous base that is uracil by a glycosidic bond, and this bond is clipped by the DNA glycosylase. So here is uracil present. So uracil is removed from the DNA backbone and leave an AP site. This is the AP site. So AP site means apurinic or apyrimidinic site, means without purine or pyrimidinic base is called AP. Then AP endonuclease cut the DNA backbone at the 5' position of the AP site. This is the 5' site and this is the 3' site. So AP endonuclease cut the 5' position and leaving 3' OH. This is 3' OH end and 5' deoxyribose phosphate end, and this is 3' OH end and this is 5' deoxyribose phosphate end. Then exonuclease cut at the 3' position. This is the 3' position of the AP site and leaving 5' phosphate. This is the 5' phosphate. This is the damaged nucleotide. So this damaged nucleotide has been entirely removed from the backbone. Next, the DNA polymerase 1 and DNA ligase fill the nucleotide gap using the intact opposite strand as a template. So this is the repair mechanism in prokaryotes. But in eukaryotes, base excision repair have two main sub pathway. One is short patch BER and another is long patch BER. That is short patch is called SNBER and LPBER. So as like prokaryote, first DNA glycosylase scan the DNA and detect the damaged bases or inappropriate base and clip the N-glycosidic bond and leaving behind an AP site. that is apyridinic or apyrimidinic base. In human, 11 types of glycosylase present. Then endonuclease APE1 in human cut the DNA backbone at the 5' site of AP site and creating single stranded break with 3' hydroxyl group and 5' deoxyribose phosphate, that is 5' DRP site. But difference is here, in short patch BER pathway typically replace a single nucleotide. So DNA polymerase beta has an intrinsic 5' deoxyribose phosphate lyase activity. So 5' DRP means 5' deoxyribose phosphate that remove 5' DRP, and the polymerase beta fill the single nucleotide gap using the intact opposite strand as a template. Then DNA ligase 3 seal the nick. So short patch BER, which replaces a single nucleotide, short patch single nucleotide replaces, but in long patch base excision repair replaces 2 to 10 nucleotide at a damaged site, especially when the lesion resists short patch repair. Then remove the damaged segment as a flap. This is the flap. So this pathway is especially active when 5' deoxyribose phosphate is resist to lyase activity of DNA polymerase beta. So DNA polymerase beta have a lyase activity. So when the 5' deoxyribose phosphate is resisted by the lyase activity of DNA polymerase beta, so then it involves polymerase delta and epsilon that displaces the downstream DNA strand, that is old strand. This is the old strand to form a flap and add 2 to 10 new nucleotide, which is then cleaved by the FEN1 flap endonuclease and that containing damaged DNA. Lastly, DNA ligase 1 seal the remaining nick at the DNA backbone. So BER is the cell precision tool for fixing small DNA lesion, ensuring genetic stability and preventing harmful mutation.